I feel like it takes a little bit longer every time I hit record for it to actually start recording. Okay, so the game plan for tonight is lectures 20 through 22. So last week's content. And there is a midterm on Thursday, which I'm sure you're probably all well aware of. But so kind of, I guess, to prep for that, tonight's session will be this, obviously. And then tomorrow night and Wednesday night are going to be review sessions. So that'll be kind of a review of everything we've done since March break. And I have, let me grab it, a Google Forms sheet. Where if you have any specific subjects, topics that you'd like me to go over, or any specific questions that you'd like to ask, this just gives like an anonymous platform to do so. So you can fill that out if you would like, or you can just message me on Teams, send me an email, whatever you would prefer. So I'm going to copy that link and I'll put it in the chat right now. And then we'll get started. Okay. So that's there if you would like to put anything down. Otherwise, if there aren't any specific requests, I'll just kind of come up with a good solid review of what I think might be more difficult and what I think is important. And I also, yeah, anyway, we'll get into it. So switching gears over to here. So starting with the theme topic of resource transport, just very briefly, how are resources transported over a short distance and how are they going to be transported over a long distance? So resource transport is critical. It's really important because, you know, I mean, it's very, I don't want to say it's like obvious, but we couldn't have life without it. We couldn't, you know, if we didn't have a circulatory system that, you know, allowed our blood to move different resources throughout our bodies, it'd be like, you know, game over. There's no way to survive like we do without some form of resource transport. So there are two main types of resource transport, obviously, short versus long distance. So short distances, resource transport can be through diffusion. Now, typically, diffusion can only be across a few cells. So 1, 2, maybe 3 cells, and that's about it. Now, that works for some organisms. Some organisms are relatively really small and simple and don't require any transport over long distances. Sponges, for example, get by on diffusion, and that works for them, which is really good for them because they're quite simple. They don't have a lot of adaptations that would allow for long distance transport. So diffusion is very, very simple, and it can only occur across a few cells, 1, 2, maybe like 3 max. Whereas if you're having long distance transport, so you need to transport, let's say, oxygen from the lungs in a human down to or up to like our brains, that's, you know, relatively, it's quite a long distance. It's only, you know, I don't know, like a foot maybe, but in the grand scheme of things, that's it's considered to be a long distance transport. And so something like that requires a transport system. So that's going to be a system of organs working together to transport those resources to where they need to be. So that system is kind of a key word, I guess, because if you think about a system, that requires, well, I don't want to get into it too deeply. Basically, okay, what I was going to try to get at with this is that, like I said earlier, sponges just get by on diffusion. Well, sponges are very, very, very basic, right? They don't have organs. They don't even have true tissue. So in order to have something as complex as a transport system, then you require true tissue of some sort. In the case of animals, I guess, but there's also, okay, yeah. There's also like plant transport, but anyway, see, I'm kind of getting ahead of myself with this. Okay, so getting right into things, circulatory systems. That is, the main type of transport system that we are familiar with, particularly in animals. So an open circulatory system is one type of circulatory system that exists. Oh, also, sorry if there's some background noise. I have my window open, so if that gets too distracting, we can shut that. So open circulatory systems, basically the key feature of an open circulatory system is that the organs are bathed directly in the circulatory fluid. So whereas, humans, we have a closed system, and so our blood is closed within veins and arteries. With an open circulatory system, organs are just bathed directly in that circulatory fluid. And so there's also, the interstitial fluid in an open circulatory system is called hemolymph. And essentially, the exchange of resources that occurs is between the hemolymph and the cells. So the hemolymph is essentially the fluid that contains the resources. So either, nutrients or different, what's the word I'm looking for? Oh my goodness, different minerals, oxygen, perhaps, that sort of thing. Hemolymph is kind of the carrier of the resources. And essentially, the cells of the organs are bathed directly in that hemolymph, and it's essentially kind of, it's like it brings, how do I word this appropriately? I kind of think of it as like, that might be a bad analogy. Okay, sorry if I'm a little bit scatterbrained this evening. So essentially, the exchange is between the hemolymph and the cells, and basically the hemolymph has to have a direct contact with the cells of the organs, and that's why the organs are bathed directly. So it's quite simple. I feel like I'm making it more complicated than it has to be. So there can be one heart, there can be multiple hearts, and the role of the heart in an open circulatory system is very similar to the role of a heart in a closed circulatory system, which we're probably more familiar with. So the heart acts as a pump. The heart pumps fluids that contain proteins, sugars, etc., whatever type of resources that might be required. So the heart's role is still the same. Heart acts as a pump. And an open circulatory system is common in arthropods, and also most mollusks. I feel like that's probably more of like just a trivia context kind of fact. I don't know that that's necessarily what I would ask on a midterm, but you never know, I guess. Okay, so in comparison, there is the closed circulatory system. And like I mentioned earlier, in this case, circulatory fluid, travels through vessels. So we have our circulatory fluid of blood that travels through our veins and arteries. So here, there's two types, I guess, of exchange that occurs. So there's an initial exchange between our circulatory and our interstitial fluid. And I'm gonna throw a diagram up here in a second that might help with this. And then there's kind of a secondary exchange between the interstitial fluid and the cells. Let me grab that diagram. Actually, there were kind of a few diagrams that I wanted to throw on here. Just looking for the right one. Okay, here's a good one. Now these are directly from like the lecture slides, so they're available to you on D2L at any time as well. But I just wanted to kind of throw them up here for context while I was chatting about it. I guess I'll throw the whole picture on. That is huge. Okay, be patient. I'm gonna grab the other one. Actually, I might hold off on that one. Well, I'll throw the open circulatory system one on here too. I wasn't going to put that originally, but I think it's, I don't know, might be useful. See, this is where doing PAL virtually actually comes in handy sometimes, because traditionally, when we've been in person, I did PAL, and I never typically connected to, I don't know why I did that. I never connected to, what's the word I'm looking for? Like the, oh my goodness. I guess, screen technology in any of the lecture halls that I was doing PAL in. So it was pretty much just me and a chalkboard, and I'm not a gifted drawer or illustrator. So sometimes virtual PAL is helpful, and then I can just pull these diagrams up super easily. Okay, so they're a little bit backwards, and I guess this should go up there. But I find this helpful to kind of visualize, I guess. So we said that with the closed circulatory system, we have an exchange between the circulatory fluid and the interstitial fluid, and then between the interstitial fluid and the cells. So essentially, actually, maybe the other diagram would have been best. Again, I'm sorry, I feel a little scatterbrained. Yeah, I'm gonna pull the other diagram up now too. I thought it might be best to bring them up like one at a time, but perhaps not. And again, there's so much content, I guess, that we talked about in these 3 lectures that it was really, really hard for me to kind of narrow it down to what I could talk about within an hour. So if I didn't talk about it, but it was in the lecture, that doesn't mean that I don't think it's important, for sure. So if there's anything that I don't talk about tonight, feel free to let me know if you want me to talk about it tomorrow or Wednesday, because there is a lot of content. So I can't guarantee that this is like the most important content either. I think it's all pretty important. Okay. Yeah, so now getting into single circulation versus double circulation. I wanted to throw these diagrams up here as well. Actually, I think what's more important than the single is talking about double. But I feel like we probably have to talk about single, at least see it in comparison in order to well, I guess compare it to double circulation. So I'll throw those up there too. This is like more detail than I planned to go into, but it's probably a good thing. So yeah, sorry if I seem really disorganized. I cut things out that I didn't really want to cut out. And so now I'm like, oh, maybe I should try to cram them in there. See if we have time for everything. So yeah, my apologies if I seem a bit all over the place. Okay, we're good now. Although we've extended the size of the document greatly, but maybe let's make them a little smaller, see if I can get them up there. Okay. Whew, now we're ready to go. So they're really tiny, but I think you can still kind of get the message. So this is single circulation, and this one over here is double circulation. And so these are strictly in closed circulatory systems. Single circulation is basically the blood passes through the heart one single time. So there's kind of unidirectional flow, passes through the heart, and then it goes around, passes through the capillaries of whatever organ. So in this example, it's through the gills and then back around. And down here, it'd be passing through the capillaries within the body, then back to the heart, back to the gills to be oxygenated, back to the body, that sort of thing. So that's pretty simple. And then in the case of double circulation, we have, that is when blood passes through two different circuits that are powered by two pumps. So this is where we have the right side and the left side of the heart. So they each kind of act as a separate pump. So it passes, the blood passes through the heart twice. So it passes, let's just start here, I guess, that we're going into the left side of the heart. And this is like the panda's left. It's our right, but it would be the panda's left, if that makes sense. So it goes into the heart and it is pumped through the heart, pumped through a systemic circuit, which I guess this was probably a bad place to start. But, okay, let's start over here. Actually, this would be better. So, right, we have the systemic circuit. So that's basically like all this blood is out in the panda's body being used up for resources. So when all the resources are used up, it goes into the right side of the heart and it gets pumped into the pulmonary circuit, which is basically the lungs. So it gets pumped to the lungs, and the lungs take the oxygen, as we know, then they're pumped from the lungs into the left side of the heart and then back into the systemic circuit, so basically back into the rest of the body. Now I know that's like a really, really quick overview, so if I've confused you further, my apologies. But I'm pretty sure there are some really good videos on YouTube about this, all of this sort of thing. So I can look for some of those later and try to post them on D2L. Okay. So originally in my question, I said, what are the advantages of double circulation? So double circulation provides more energy. So typically organisms that have this ability to undergo double circulation can be more active generally. I mean, the example here is a panda. We know they don't really do a whole lot, so that's kind of a bad example. Also supports higher blood flow, or maybe a better way to word that would be higher blood pressure and higher flow rates. Okay, so again, I wasn't intending to go over that because I thought we wouldn't have time, but I do think it is pretty important. So if I did a poor job with explaining, let me know and I can try to address any questions that you might have. Okay. So then getting into xylem and phloem. What are xylem and phloem driven by? So we've already talked about xylem and phloem quite a bit. We did get into more detail in this lecture than previously, and I do think those details are probably important. But I think the most important part to take away from what we talked about were the different processes that drive these flows. So the xylem flow, if you remember, xylem is responsible for the flow of water, mainly, is what xylem is associated with. And so it is driven by transpiration through solar energy. And I'll bring a picture up real quick. Phloem is the more confusing one, I would say. More, maybe not confusing, but complicated. I'm going to turn a light on. I didn't know there was a phone there. It made a beeping sound. I touched it. Sorry about that. So yeah, phloem is a bit more complicated. And so the process of phloem flow is through translocation, which is driven by, metabolism. And that requires ATP. So it's an active process that requires ATP to move phloem around. So let me grab a lot of these things that we talked about. I found were very helpful when you accompanied them with a diagram, in my opinion. So yeah, the xylem flow by transpiration, that's essentially just the, if you remember, xylem is basically getting water from the roots of the plant. Let's just go with like a tree because that's, you know, we're all familiar with trees. So xylem is responsible for getting water from the roots because we know that's where trees get their water from. And then basically sucking that water up through the trunk of the tree and out into essentially all the smaller branches and stems. And so that's driven by solar energy. Essentially, you can think of it as evaporation is a big kind of portion of that. And actually, let me throw the diagram up here first. So all of these diagrams are from your textbook too, I believe, not just in the slides. Let's shrink this down a little bit more maybe. Okay. So this kind of shows how xylem and phloem work together, which I really like, which I think is important. Yeah, so there's the xylem. It essentially is pretty straightforward, just going from roots down here up to the shoots. And that's driven by solar energy, like I said. And then everything that takes place in phloem is a little bit more complicated. So phloem is going from high to low potential. And so if you remember my pretty bad analogy, or not analogy, I guess, I don't know what you would call it, my memory trick for phloem is that it kind of, if you squint your eyes enough, looks like the word maple a little bit. It has almost all the right letters. So phloem is responsible for moving sugar throughout the plant. And so you see here that there are all these little molecules. Oh, sorry. Excuse me, I don't know where that came from. Are sucrose. And so the sucrose can move into this phloem tube. And essentially, it's moving from high to low potential, which I just said, which makes sense because if you have a lot of something at one end and a little bit of something at another, it's just naturally going to kind of spread out throughout that area. So sucrose tends to kind of flow down this way into the zinc cell. So where xylem typically moves things from the roots to the shoots, that's sort of the tagline, I guess, for xylem movement. The sort of tagline, I guess, for phloem movement is typically from source to sink. So the source here, we're getting sucrose from a leaf because we know leaves are where photosynthesis takes place. That's where the sucrose is produced from solar energy. So that's our source, is the leaf. And it's moving down to the sink, which is the storage root. So it moves from the leaves to the roots, and then the sucrose is stored there. And so water as well kind of moves with it, and then that water cycles through and helps to move, or I guess it doesn't help to move, but it gets moved back up to the top through the xylem. So it's sort of a cycle. I think that's all I really wanted to say about that as well, is that it's very cyclical, and to point out the from the roots to the shoots thing with xylem, and the source to sink with the phloem. But if there are any questions at all, please let me know. Because again, it's a bit heavy, I think, and there's a lot to cover this evening. I found these lectures to be really, really content heavy. So if I'm moving quickly, let me know and I can slow down. Okay, so next thing, what are the two media where gas exchange can take place, and what are the qualities of each? So this sounds a bit odd, but basically the answer is quite simple. So gas exchange is something we all know about, right? Animals, for example, we inhale oxygen, we exhale CO2. Plants take in CO2 and they give off O2, that sort of thing. And so two media where this gas exchange can take place, one is air. And the other is water, because we know, gas exchange takes place in aquatic plants as well. It takes place in aquatic animals, so fish have gills, that sort of thing. Okay, so comparing the two sort of media together, air is a low density medium. So it's not very dense compared to water, right? I mean, if you think about moving your hand through air versus moving your hand through water, water is going to be a lot more dense. Air is also a lot less viscous. So viscosity is like thickness. I always think of like molasses when I'm thinking of viscosity, because that's a very, very viscous substance. And then in air, there's also lots of oxygen. So in air, our rate of gas exchange doesn't have to be perfect. It doesn't have to be super efficient. If we, it has to be efficient to some extent, or else we'd suffocate, but we don't have to be like expertly skilled and super, super efficient that every breath is bringing in loads of oxygen. Oh my goodness. There we go. So water, on the other hand, is dense in comparison. It's viscous in comparison, and it has much lower levels of oxygen. So here, the gas exchange needs to be efficient. Oh my goodness. There we go. So yeah, those are basically, I think that's important. So I wanted to talk about it quickly. Okay, so the next thing, key features of respiratory organs. So respiratory organs, there are a few different types. We would probably be most familiar with lungs, for example, that's what we have. But also gills are a really, really common respiratory organ. And basically one super key feature of these respiratory organs is that they have typically an enormous surface area. So our lungs, it's mind-blowing, the surface area that those suckers have. And also they tend to be pretty thin, which allows for, oh my goodness, allows for exchange. So in terms of why you would want to have this enormous surface area, it's going to allow for more gas exchange. The more surface you have, the more exchange that can take place, and same with it being thin. That just allows for exchange to take place more easily. So let's start with gills. Gills are an outfolding of the body surface. Okay, well, that's a word, so, oh my goodness. I don't know what happened there. There, word likes that better. And essentially, the deal with gills is that oxygen diffuses from the water to blood in the gills. So basically, their part of the body is folded, to the outside so that water can run through it, and there is dissolved oxygen within that water. And so that oxygen that's dissolved within the water can diffuse from the water through the gills into the blood supply running through the gills. That's essentially how gills work. So yeah, gills, like I said here, are pretty thin, and they generally have a large surface area, which makes them more efficient. And we said earlier that organisms in water need to have really efficient gas exchange, so gills are pretty good at doing that. In comparison, let's talk about lungs. They are an infolding of the body surface. Our lungs are, folded inside of us. And basically the key to the lungs are the alveoli. So our lungs are a very kind of sponge-like tissue. I don't know if anyone else is like this, but recently I've seen a lot of TikToks going around on my For You page that people are like shocked to find out that our lungs are not hollow. They thought they were just like full of air, but they're kind of sponge-like, and they have these alveoli, which are essentially, they sort of look like grapes. They're little like mini sacs of air. And so we have remarkable surface area within the lungs. They're not just, solid chunks of flesh, right? They have lots of like small branching structures that give them way more surface area than you would think they would have. Yeah, and so the alveoli are a key sort of unique feature of the lungs that I think are important. We also briefly talked about tracheal systems. So essentially, these are common in arthropods, I believe. The example that we looked at was like a grasshopper, I think. So these types of organisms that have these tracheal systems have these kind of pores on their body called spiracles. So air is able to enter through the spiracles, and then it can travel through these tracheal systems, which are basically a system of increasingly smaller tubes. So you get smaller, or you start out big and then you get smaller and smaller. And again, that's going to allow for a lot of surface area to go from big tubes into tiny, tiny, tiny ones. Yeah, okay. Moving along. Gas exchange regulation in land plants. I wanted to show a diagram for this one as well. Essentially, this process is regulated by stomata, which are, again, tiny pores that are controlled by what are called guard cells. Oh, okay, never mind. Sorry, I have a Teams notification. I thought it was the chat, but it was something unrelated, so sorry about that. Okay, so the stomata. I'll bring up the diagram really quickly. I think it is valuable in understanding. And I think it's really interesting as well. All right. Let's size it down a little bit, maybe. Okay, there we go. Could go a bit bigger. Okay. So essentially, the stomata itself are the pores. I believe it's stomate if it's singular. So the hole itself is the stomate, the holes are the stomata, and this is depicting kind of the guard cell that's situated around it. And so the guard cell does exactly what it sounds like it would do, sort of guards the pore, and it kind of can open and close the pore, allowing for gas or water to flow in or out. And so essentially how this kind of goes down is that all of the little red dots in the image here they represent a potassium ion. So basically you can see in this example, we're starting with the open stomate. And when we have this accumulation of potassium ions, what happens is that the water potential decreases. And so water is able to enter the cell. So when we have this accumulation of potassium ions, water enters. And you can kind of think of it as, I guess, I'm trying to think, did we talk about that this year or did we gloss over it? Back with, in Dr. Patton's section of Bio 1001. I'm pretty sure we talked about it. Essentially, if you have the whole goal of any biological organism, I guess, is homeostasis. It wants things to be regulated. So if we have a whole bunch of potassium within this guard cell, then water is going to flood the guard cell to try to balance it out. Because the plant would prefer for things to be sort of neutralized. So with all these ions accumulating, water is going to flood into the guard cell to kind of try to balance the distribution. They don't want a whole lot of potassium. And so when all that water rushes into the guard cell, the cell becomes turgid. So it becomes really stiff, and that opens the stomate. So they're really rigid, and they kind of push against each other and open that pore. And so when that pore is open, then gas exchange can kind of freely happen within the land plant. So when there is accumulation of potassium ions kind of outside of the cell, so there's I think, what's the word, an efflux, I believe, of potassium ions sort of, they're all on the outside of the guard cell. So there's very few inside. There's a large accumulation of potassium ions outside of the guard cell. So in that case, the guard cell has too much water in it compared to the amount of potassium within it. So it's going to expel water. So water is going to leave the guard cell, and that's going to leave the guard cell flaccid. And so it kind of closes in on itself, and it closes the stomate, so that gas exchange is basically cut off. So that is how that sort of works, that regulation of gas exchange in land plants. So water and potassium play a really big role in oxygen and CO2 sort of entering and leaving. Okay. Actually, it's funny that I mentioned that earlier about me being able to just copy and paste diagrams in, because I remember I covered this in my very last session before COVID struck and we went online in person, and I was trying to draw this on the chalkboard and it was horrific. So yeah, this is a lot nicer some days. There are pros and cons to everything. Okay. Moving along into lecture 22 on growth. So what is growth? That seems like a little bit elementary, but it's important to have like solid definitions, I always find. So what is growth, and what does it look like in plants, animals, and unicellular organisms? So growth, you can think of as irreversible increase in body mass. So let's start with, what do we want to start with? Let's start with animals because it's what we are familiar with. So growth in animals. Oh, okay. Ooh, I was looking at the wrong thing that I had written down, and I said, oh my goodness, that doesn't make any sense. But this makes sense. So we are considered to have, oh my goodness, determinate growth, which basically means that we grow fastest, oh my goodness, as an embryo. There we go. And we basically stop growing when we reach like a juvenile phase. So humans, for example, we grow a lot as babies. And even, the first 5 years of a kid's life, it's like, they grow from being super, well, you're not that tiny when you're born, but tiny little babies to like an actual, person, a 5-year-old kid. And then if you think of that kind of that period of growth between like 5 to 10, there's still like a lot of growth going on. Probably not as much as going from a baby to a 5-year-old, because that's, really extreme. 10 through 20, there's like, well, I guess there's quite a bit of growth that goes on. But again, probably not as much, right? And then by the time we hit around 20 or so, growth is done. We're not going to grow anymore. I guess this is where we could talk about the growth being an irreversible increase in body mass, because growth in humans, you can think of it as, growing taller. I've definitely grown wider since quarantine occurred, but that is not considered to be an irreversible increase in body mass, so that's technically not growth. But I haven't grown any taller, I don't think. I haven't grown any taller in a few years, I don't think. So technically I haven't grown. But that is what basically an irreversible increase in body mass refers to. So yeah, with animals, we have a determinate growth. We grow really, really fast as embryos. We grow fast when we're, kids, and slowly it starts to kind of peter off how much we're actually growing until we eventually stop growing. And that's generally true for all animals. Whoops. Oh my goodness. For plants, they are kind of the opposite. So they are considered to have indeterminate growth, which basically means that they grow throughout their life. So plants never really stop growing. In their sort of youth, I guess, they can grow a lot more quickly. And as a plant kind of reaches senescence, so it's like end of life, its rates of growth can slow down. So for example, personally, I'm a big plant gal. I've brought home a lot of plants that, when they're just little babies, they shoot right up, and I'm like, queen plant mom, I think I'm doing such a good job with them. And then they plateau, and it seems like they grow maybe a couple centimeters a year. And that happens. But they always do continue to grow. Just as they get older, it's going to start to peter off in speed a little bit. So they grow throughout their life. There's no point that they hit and they just stop growing. Yeah, it might slow down, but it's never going to just cut off and stop. They always keep on growing. Maybe mine don't grow super well because once the novelty wears off, I forget to water them. That would explain it. So those are plants and animals, unicellular organisms. So think of tiny prokaryotic bacteria, for example. Their growth occurs, oh my goodness, before division in mitosis. So think back on what we learned about mitosis in Bio 1001. We had the G1 and G2 phases where growth occurs. Oh my goodness, what's the word? Replication of all of the chromosomes within the cell, that sort of thing. And also, when you hear the word growth in the context of a unicellular organism, so in the context of a bacteria, it's usually referring to colony growth, which is not, growth of one individual bacterium, but it is, a colony of bacteria that can double every 20, 40 minutes, whatever species you're working with. Yeah, so it usually you wouldn't discuss growth in the context of one single bacterium. It would usually be talking about colonies of bacteria doubling as time passes. Okay, so then what is development? Sort of similar but different to growth. That is when we have a change from a simple to a specialized form. So in animals, that looks like, and again, this is a really quick kind of blasting through it, because these slides, these lectures were pretty content heavy, but we basically go from a zygote, which is just sperm, egg, stuck together. That's a zygote. Very, very simple. And we develop into a blastula, which is a hollow kind of ball of, I think, 16 cells. And that develops into a gastrula, which is basically, more cells. And what's important about the gastrula is that it can have 2 or 3 layers. So that is when development occurs, and kind of there's the distinction of, is this organism, is this animal going to be a diploblast or a triploblast? So we talked about that with Amy a few weeks ago now, I guess. But yeah, unfortunately, you can't just erase that out of your head. just because you had the final, which sucks, but it came back. So yeah, the gastrula can develop to have 2 or 3 layers. So it will always have the ectoderm and the endoderm layers. And so the 3rd layer is the mesoderm. So not every animal has a mesoderm. So this is kind of where the first distinction takes place. Plants, on the other hand, their development can kind of be categorized into 2 groups. So they have primary growth, which occur in the apical meristems, which I want to get back to real quick. Okay, yeah, maybe this is a better way to kind of start this. So meristems are, you can essentially think of them as stem cells in plants. That's how I kind of keep them straight in my head. And so there's 2 different types, and so they, undergo 2 different types of development or growth. So the apical meristems sort of look after primary growth, which increases the length. So that the apical meristems, you can find them at, the tips of the plant, and they are going to help the plant grow taller. Actually, let me bring this over for a little example. This poor little plant, I've had it over a year. It just sprouted these 2 new leaves quite recently. Those are the first new leaves it's had since I got it over a year ago. It looks kind of rough because I haven't watered it in a while. I like to think I take care of my plants. I really don't. But yeah, so the apical meristems would be, the meristems which these leaves resulted from. So they came from the top here. So you probably really can't see, but there's like just the stem, and then it gets more and more narrow as we get to the top. And then at the very top, there's this itty bitty, very thin little area that's, very short. But it wasn't there a few weeks ago. So that's primary growth. It made the plant get a little bit taller, and then quite a bit taller once you factor in the leaves. So that's primary growth. 1 second. And then we have secondary growth is through the lateral meristems. And secondary growth increases width, or like the girth of the plant. So that would be, let's turn it around. There's less leaves on this side. You can probably see, you can see the dust on my pot. I'm a terrible plant mom. So you can see probably here that this area of the stem is thicker than, say, this area of the stem. And so secondary growth increases the width or the girth, and so that is the type of growth that makes the plant grow wider. So probably if I took good care of this plant, up here would be more wide, but I take pretty poor care of my plant, so it's struggling. Yeah. But basically that is primary versus secondary growth in plants really, really quickly. Okay. And this is the last thing that I wanted to briefly touch on. I know we only have a few minutes left, but I think we'll get through it. And still have time for a few questions at the end if there are any. So, how does the environment help determine the phenotype of an organism? So we previously have said that the phenotype comes directly from the genotype. So it is true that the body plan is encoded genetically through the genotype. So thinking in the context of humans, for example, if we are genetically predisposed to be really tall, then we're genetically predisposed to be tall. There's probably not a whole lot of things that are going to change that. But this, I think, is it makes a lot more sense in the context of a plant. So bringing back this good plant here. Perhaps this plant was, actually this is a really good example, was encoded genetically. Perhaps it had a genotype to grow really tall and to grow lots of beautiful stripy green leaves. And maybe, you know, that's what it was supposed to do. That's what its genotype was for, was to grow tall and to grow lots of leaves. However, the environment that it ended up in is Maegan's house. And that's not a prime environment for growth, apparently. Probably would have done a lot better if it stayed at Scott's nursery. Because that environment probably has more sunlight, more water, more, you know, rich soil. I haven't changed this soil since I've gotten it. And I don't think I've given it any nutrients or anything. So yeah, the environment is not too great for this plant. So maybe it was supposed to be this wonderful tall plant with lots of leaves, but the environment that it's in is not super great. So the environment has determined that it's actually going to be a little wee short plant. So the phenotype might have been, it was supposed to be a nice tall plant, but because it hasn't been given the things that it needs to thrive, because I'm a neglectful owner, the phenotype has been impacted because the environment is not ideal. So things like nutrition. I never give it fertilizer. Stress. I don't think it's, well, it's probably in constant stress actually. The amount of light it gets. Honestly, it doesn't get a lot of light where I have it. It would get a lot more light in, you know, a windowsill. Water. It's pretty prone to drought when I'm looking after it. So, and then other things like competition, predators. There aren't a lot of competition or predators in this example, but you can imagine in the wild, competition is a real thing. So maybe you have, let's say you have a bunch of saplings, little tiny trees, and one of them is supposed to grow super tall, but maybe it gets a late start in its growth. And so all the other saplings around it have already grown above it. And because of all that competition, all of the competition around that one little, they are going to get more light just because they're taller. And so the other plant is going to constantly be in shadows and not receive as much light, so it won't probably grow to its full potential. So yeah, the genotype definitely plays a role in the phenotype of an organism, but the environment can also impact it. So I've really exposed myself with being a bad plant owner this evening. But I deserve it. I used to be very good with my plants. I've slacked off. So yeah, hopefully that makes sense. If you have any questions at all, feel free to let me know. And again, access the Google form if you would like to send in any anonymous requests for kind of content that you'd like to see covered during my review sessions this week. Yeah, I'll stop the recording, and if there are any questions at all, just let me know.